Hello. Yeah. I have one quick question for all the speakers, especially my mentor Don. As you already mentioned, that three failed clinical trial disease use ASO. One is Toxin 2, another one is the Huntington, a third one is the C9. They all belongs to polyQ disease. So is that a coincidence that ASO cannot treat the polyQ disease? Or
So I don't think so, but and I mean they're not all polyQ. One, they're all repeat diseases,
yeah,
but the ataxin one, I think was just it's a complete drug failure. The suppression of ataxin was extremely modest, and I would have so I have no that was run by Ionis Biogen, and I have no part to that. But I looked at the final data; they barely suppressed ataxin, so I think it was just a complete failure. And now with regard to what Dr. Lee just described, that it may well be that the drugs chosen, the ASOs chosen, were just simply not effective enough. And this delivery method that you just heard from Ractogen, and I'm going to say it in a much, in a very, very direct way, they're gluing an ASO onto an siRNA. And they're now making a quite big drug. But the question, already the ASOs are 15 times the size of a typical drug and heavily negatively charged. But by using the pathway of uptake, the ASO uptake has really made those siRNAs more effective. So I think we should be very open-minded that maybe this is and delivery has always been a key issue. It remains a key issue for neurodegenerative disease. So and improvements should be welcomed. And there I think the failed Huntington trial, the failed C9 ORF72 trial, I think there's the team, the broad team does not agree on the basis for the failures. So, and I'm sure that the audience here is going to have their own views as well about why they failed. Certainly, the drugs were not as active as you would have liked, and there was some evidence that could be interpreted as the drugs had their own toxicities, and so dose dependent. That is indeed what Roche concluded in terms of the redo of the Huntington trial, where they're starting way earlier, patients way earlier in disease, but also with a lower dose of the drug because they were afraid that actually that they were seeing drug-mediated toxicities. And these ASOs, actually, I think for the in Lorem and for the other very rare disease examples, the issues about toxicity of those ASOs, those are very real. The Ionis examples that I'm familiar with, they've made and tested 1,000 different ones every time they went to trial. Their competitors have frequently shown that they do know how to make toxic ASOs. And Ionis was kind of quite pleased that actually, and I should have mentioned in my talk, and then I'm going to shut up. So I meant to mention, especially because representatives of ALS Network were here today, the founding ASO that went into the human nervous system, Ionis was not committed to that project at the time. Their CEO was opposed to working in the nervous system, and the preclinical toxicology of the first drug that went into man was not paid for by Ionis. It was paid for by ALS Association, Golden West ALS, and by MDA. It was Tim Miller and I wrote applications to both of those organizations to provide $1 million of funding, a large amount of funding from each of those organizations in order to enable the first in-man trial. And because that was safe, that then enabled that was the birth of the SMA program. It was the birth of Ionis's focus on neurodegenerative disease, because they really could deliver them broadly and safely. But they work very hard to find ones that are sufficiently safe.
I might just add that in the case of C9, I think that the disease mechanism is more complicated than some people thought originally, and I don't think there was a lot of like listening to the cells and what the cells say about when you treat with a certain approach, which I think is a good lesson for just a lot of the things that we're doing now with these, N of one studies or other, ASO studies. And it's really important to test that therapeutic mechanism and make sure that's the right one.
So thanks, everyone. This has been really inspiring to think about. Looking at the heterogeneity and all the preclinical models that you guys have all presented and talked about, there have been a couple paths people have chosen when looking at these CRISPR mediated, so you have an isogenic control versus patient derived, so you can look across a panel. Kind of tying this to oncology in a multiple hit model. So you need some sort of genetic, then you need 2 or 3 other hits along the path to actually get disease. Do you feel like in these neurodegenerative settings, we need to be dealing with panels of patient-derived as opposed to 1 or 2 well-characterized models where we've CRISPR engineered in a diverse set of mutants?
So certainly many definitely think that, and I have to say I'm not one of the primary advocates that you need 1,000 in order to be able to get an answer. I'm actually worried about a different thing, and I'll let the others talk to that question, but then I add to your question, which is that and I know this is going to be deeply unpopular at iXCells. So the idea we had demonstrated, at least for disease in mice, that SOD1 is a ubiquitously expressed gene, and the cells that drive rapid disease advance are not the motor neurons. They're the microglia and the astrocytes, their synthesis of mutant SOD1. So now if you're taking an isolated motor neuron, is that going to be adequate for, are we reverting to now assuming that the disease is cell autonomous from mutant action within individual cells? To me, that's actually, it seems to me to be unlikely to be true. It didn't say it doesn't play an important role, the synthesis within the motor neurons, but it does kind of, so my question to Justin or others would be, how are we going to, are we going to consider other contributions from those mutants in other cell types other than the primary motor neuron?
Absolutely, we're going to have to.
Yeah, I think that's a huge part of it. It's way more complicated to incorporate those cells in my experience, but it's really important. I think for things like ALS, there's an opportunity where certain patients may harbor genetic lesions and genes that are highly associated with ALS, but maybe not. There's not a strong example of many families, where this mutation is separating with disease, but likely it may play a role in disease. And I think there are many people initiating efforts to make ASOs for those genes, and in hopes that it might help the patients that are just a subset of ALS patients with mutations in certain associated genes. And so there, I think it could be useful to see if indeed, this ASO strategy could work for those patients. This would just be a way of trying to get at the sporadic population in like some subsets.
Thanks. I as someone who 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 is not a neuroscientist, but has been 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 following the 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 literature closely, and 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 who thinks about this in in terms of in terms of developing 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 technology. I I I don't think that we know the answer to the question yet, but but one reason that 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 I find the work that that Justin has done, and and others like like Alyssa Coyne and John Fauci in particular, that 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 we now have several several independent examples of of these of these of these heterogeneous patterns showing up in 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 the in the in the in the transcriptome, and and I think that that a big question that we need to answer is 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 how much can we learn from that, and 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 that is why with why with why with Project Mosaic, at least, we in the early phases we want to know are 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 those patterns reproducible in the same mother neurons, and if so, is is that indicative of the underlying biology, and then and then if it is, we can begin to answer more more complex questions around well how much can these models tell us about 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 how drugs might work in in different subgroups, and to your point, what why can't these 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 models 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 tell us. Sorry, and then that is where we will probably need 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 more sophisticated more sophisticated co culture, and eventually possibly 3D or 3D organized, but but we have to we have to certain certain somewhere.
My question is for Justin. So I am very impressed by the results you showed that you did a RNA CRISPR-I library screening to identify two target genes, which knocking down both either of them showed a protective effect. So I'm just wondering if you have gone to the other direction, so try to screen like overexpression library to see whether overexpression certain targets also may have a protective effect.
Yeah, thanks for that question. We didn't directly do that, but we do have genes that are sort of negatively enriched when you knock them down. So they sort of kill the cells more when you knock them down, and so those might be candidates, for overexpression. If maybe some of this RNA, small saRNA technology, can work to upregulate genes, I think that would be powerful if that could work. And so I'm kind of curious to see how that develops. Yeah.
Yeah, and maybe if I can return to the former question as well. So maybe where a broad, large number of samples, one place there are about 30 diseases for which there's a nucleotide repeat that's causative. Maybe the most famous one is Huntington's disease, but also for C9orf72 in ALS. And there, the different populations, right? The C9orf72 expansions are almost absent from China, but they are the largest cause outside of China. They're the largest known genetic cause worldwide. And there, there was a long discussion at the nucleotide repeat meeting about the partial penetrance of the C9orf72 mutations, with multiple authors identifying in different populations that it's only about 25% penetrant. So if you inherit the expansion, you have a real chance, but a 25% chance of developing disease and a 75% chance of not developing by age 80 in any ALS-like phenotype. And so there, maybe, so maybe I'll even turn that into a question for Justin or others. The question, if you do look at those, are there sequence variants that can now be protective? And is it in the in vitro making these neurons? Do we see differences in the phenotype in those?
Yeah, that's a great question. I don't think we've done enough lines carefully enough to say if there are differences. I think there's that TMEM106B gene that might be a good example of with a certain variant in that gene, you strongly protect against getting FTD with the C9 repeat expansion. Maybe there are things like that for the ALS side. Yeah, I totally agree.
So that's definitely an area where many different cell lines, especially if they were collected like in ways in where they're really comparable, been produced comparably. Actually, I would also then I would really strongly advocate that that none of us should be arrogant enough and to think that we know what the right way to do it is, and nor should we think that the perfect should not be the enemy of the good. And so the strategies Justin now has proposed, three targets. Those are good. Those are good targets. I proposed one today as Stathman, where I think we can do good. It's not by no means perfect. It's not putting TDP43 back, but it's putting back one of the proteins that's required for maintaining innervation. And so this is and there'll be others. And so I would really broadly support the idea that different ideas is the right direction, and don't let and Sherry's gonna kind of dislike part of this. The idea that organizations want to band together to identify the best idea that a whole broad spectrum of folks will think is an okay idea, that's probably not the right way to go. You want to see that the discoveries, the true discoveries, are going to come from unexpected directions, because otherwise they wouldn't be the grand new discoveries. So I would really want to see that a broad spectrum of folks that can defend their strategy. It won't be perfect, but it will be it's another shot, and we need as many of these shots on goal.
I just want to say that those are the exact ideas we hope will come into apply, and that people like you with running labs who have scientists who may have a real out of the box idea, you know, this RFP gives them a place to get some funding for that. And you're on the committee to help say, hey.
So again, while today's meeting, and I could see it's now just accepted as an obvious fact, the ASOs were initially not at all accepted, including at Ionis. The idea that they could work in the nervous system, that was not believed inside Ionis, and the investment was made by ALSA and then MDA to develop that, and we did not understand that we could deliver these things broadly. We did not think we could. We thought we'd had to use continuous flow. We had no idea we could get 4-month efficacy periods, and as you saw from Ractogen, even better for this fusion of an ASO onto an siRNA. It's just like you have to do it and then critically assess it, of course. But then you applaud success. You're disappointed by failure. But that I think is the real process that has enabled us to get to ASOs are going to be used broadly in the nervous system. We heard it all day, right? I didn't include today. I do frequently, but I didn't today. The original we sent that work for publication, including showing that it was effective in non-human primates. And that was editorially rejected as no contribution by Nature Medicine. It's like then Huntington, we showed we could get partial disease reversal in 3 Huntington model mice. And they said, well, that was nothing either. It's just, and in the end, I got that editor on the phone, and he said, well, he admitted that showing that you could do it in non-human primates, it was remarkable. But then he concluded that it was no intellectual contribution. Because you knew if you turned the gene off, it would be good. And then he said, ASOs will never be used in human therapy.
We're running a little bit late, but I have one last question. Sorry, I took the last one. So for Dr. Lee, are you still there online with us?
Yes.
Okay. So because after hearing your presentation, I am again confirming my belief that the technology can be, because it's targeting, they have very tissue specificity, and also the delivery efficiency is much higher with this, conjugation with ASO. So I think this modality can be very beneficial to some of the ultra-rare neural disorders. So as one of the biggest audience today. So I'm just curious, do you have any strategy how to utilize your technology to help with those ultra-rare patients? I know we have a little bit discussion with Amber-free, SL661, but it seems that we don't have very clear roadmap and path how to move this forward. Partially it's because of the politic barrier, and also part of it because of the financial barrier. So I'd like to hear about your thoughts.
Oh, yes, of course. That's a good question. The last year at the Boston CNS delivery meeting, I talked to some people from the ultra rare disease organization like Everyone Medicine, and so propose to them that if they are interested in using siRNA for ultra rare disease, and they can use the scattered delivery system to quickly deploy the therapeutic, because it's just like a plug and play, right? Very quickly. And actually, we are doing this in collaboration with the very famous patient, ALS patient, China, in China. We are now designing and the screening allele-specific siRNA for some mutation identified in a certain patient. Yeah, it's quite feasible.
All right, so I guess we are closing. We are gonna have a closing remark by our one of the speakers. So can I ask? Thank you so much for you guys. So I ask you to stay with us a little bit, 5 more minutes. As a tradition of our events, we always think this kind of events and initiatives should be patient focused. So the patient's voice matter a lot. So we always ask one of our patient advocates to close our events. So this year, so it's gonna be Michelle.
Thank you so much. I just want to take a minute to highlight the need right now for cross-disease collaboration, especially right now. It's obviously an extremely uncertain time for the rare disease space, both in terms of federal funding and potential changes at CIRM, as well as ongoing challenges on the industry side. So it's really, and we all know that when funding dries up, rare disease programs and grants are the first thing to get cut. So it's really important for us to all come together and put a spotlight on rare diseases and bring attention to the needs and the opportunities in this space. We need to get the message out in the mainstream that rare is not actually rare, bring attention to these devastating diseases, but also the historic and transformative treatments that are on the horizon. And I'm going to put in a shameless plug for my Zebra Run for Rare, which is not actually planned yet because the zoo is not cooperating very well. But I'm not letting the dream die. As one of the world's leading biotech hubs and the home of the world famous San Diego Zoo with our real life rare disease mascots, I think San Diego is uniquely positioned to host a really large scale, high profile event that can raise funds and awareness for rare diseases. Neonway and I and some other groups were actually in talks with the zoo last year, and we even had the mayor's office involved, but in the end they kind of told us that, well, we can't do any more runs unless it's for organizations with close ties to our board. So if any of you happen to have high-level connections at the zoo, please let me know. Oh, there's also going to be a great event on Rare Disease Day downtown at the Brigantine next week. So it would be great to see many of you there to continue these connections and collaborations. Finally, I just want to say thank you to iXCells for hosting this event. Thank you to everyone and all of you online as well for being here today. Thank you all for caring about rare. I have 100% confidence that life is not always going to be like this for my son Tristan and for our family. And I know that by working together, collaborating to accelerate progress, we can make a difference for hundreds or thousands or millions of other kids and families like mine. So thank you all.